Okay, everyone. So welcome to the first webinar of FAIRDOM's Data and Model Management webinar series. And special thanks to all of you who made an extra date in your diary after our teething difficulties last week. We're really happy to see you all back. Now, in case you're wondering, I'm Natalie Stamford and I'm the FAIRDOM Community Lead. And as you may know from the email, FAIRDOM is an initiative to support researchers, students, trainers, funders, and publishers. In fact, anyone interested in data and model management, in better management practices. We offer software, advice, expertise, and training, which help make managing your data and models in life sciences easier. And this webinar series is part of that help. We want to bring interest and knowledge to the community about data management. If you want to find out more about FAIRDOM, you can check out our website at www.fair-dom.org, or you can follow us on Twitter at FAIRDOM_EU. And if you have any questions regarding FAIRDOM, you can either reply to me from your webinar invite emails, or you can get in touch with the wider community at community@fair-dom.org. And today we're here with our special guest, Stephen Wiley, and he's from Environmental Molecular Sciences Laboratory at Pacific Northwestern National Laboratory in the US. So it's actually 6 o'clock in the morning for him here. That's how excited he is to bring us some good news on data management practices. Morning, Stephen.
Yes. Oh, I'm here. Can you hear me okay?
Yes, we can hear you.
Okay. Ready to go. You're going to give me the control of this, right?
I will give you the control in a minute, Stephen. Are you doing okay this morning?
I'm doing just fine, thank you.
That's good. Okay, so Stephen's a pioneer of computational biology, and he published one of the first computer simulations of cell dynamics. He's also one of the few researchers in the field with extensive experience of hands-on science, modeling, data management, and technical widgetry. So we're very lucky to have him with us today. As a competent all-rounder, we thought he was the perfect person to start the series, and he is going to talk today on future-proofing your data, working to ensure that your work will survive the connected world. Now, if you have any questions during the webinar, you can raise your hand and we can come to you at the end of Stephen's talk. Or you can post in the chat just at the bottom, and then we can bring up your questions later on when Stephen's finished his talk. The webinar is going to be recorded, so we'll make it available offline, so you can also access it afterwards. So I'm just going to hand over to Stephen now, and we'll start.
Okay. There it is. Okay, can everyone see the slides now? Let's see.
Yep, they're up, Stephen.
Okay, there it goes. Okay, what I'm going to talk about today is sort of a concept that I have about what I call future-proofing your data. Because what's happened is that things are really changing a lot, especially in the biological sciences, with how we take data, how we process data, how we use data. And I'd like to use the perspective of the standard scientific way that we on how we normally do science. Typically, when you do an experiment, science is always linked theory, experiment, and analysis. That is, you start with an idea theory, you do some experiments, you get some data, then you basically process it, and this basically leads you to do some new experiments. Now, in years past, all of this was done basically in a single investigative laboratory. It's because they basically, everything was done, and the scale of the science was actually fairly large. But as time went on and things got more and more complex, there became an obvious need to change this paradigm. So instead of just looking at very small theories and ideas, now we look at complex metabolic and regulatory networks. And to be able to analyze such a complex system, we have to go to advanced high-throughput technologies. These technologies, like gene sequencing, proteomics, metabolomics, generate an enormous amount of data that we then have to analyze that then goes back to the theory. So in essence, what we're faced with now is the challenge of taking a large amount of data and integrating it into some coherent way. And unfortunately, the way that we handle data, process data, and share data has actually changed, has not changed, and so we're stuck with things sort of the way they were many years ago. Now, the standard way of communicating ideas, concepts, and data has traditionally been the scientific publication, in which you have a title which in essence tells everyone sort of what you were doing, an abstract which in essence summarizes what you did, and then you basically talk about the what other people did, how you did your experiments, the results, and the discussion. And in essence, the data that you actually collected to do the experiment, you basically summarize parts of it into a figure, tables, and such. So in essence, a publication is not actually the data itself. It's actually just your impression of what you think the data actually means, and you throw just a smidgen of that data out there so that people can sort of say, see, I actually did the experiment, and this is how I interpret it. It's to give confidence in what you've done, not necessarily to share the data. Now, what's happened in the meantime is instead of having a little bit of data, now we have a lot of data. And so now the publications that we're submitting these days have less and less of what we've actually done. So much so that in the last couple of papers that we did, we only talked about one very small fraction of one very small issue that we had actually found in all the data that we generated. So what's happening now is that the original scientific publication is no longer really an adequate way of communicating what we're finding and basically enable science to be done in this new systems level way. So one of the biggest concerns that scientists have nowadays is this idea about the data deluge. And this is just a figure from a Nature paper in 2009. Of course, it's a lot worse than this now, in which we're getting more and more data, and we really don't know how to handle it. So very rapidly, biology is becoming a data-driven science. The ideas we had yesterday have become supplemented by the need to predict on the individual level. Now, once everyone has their genome sequence, and once we know exactly the molecular determinants of disease, then we basically are going to be moving towards personalized medicine. And this requires a lot of data, integrating lots of data, and understanding data to a degree of complexity that we've never seen before. And actually, this is a problem that I became very aware of early on, and I took very personally, because what I had done is I had built up a scientific career from my idea, but not that much data. As I have transitioned to more high throughput technology, and it takes me longer and longer to sift through it, I realized that I can only publish interpretations of a very small amount of the data that I generate. So what do I do with the rest of the data? Dump it, put it in a public repository where it can be ignored with most other public data? Or perhaps I should become a data scientist and spend all my time building databases and forget about my science. But there's only so many hours in the day. And this was really the frustration. I knew how important the data was, but I actually wanted to do my science. I didn't want to have to spend all my time trying to play with data and try to find out what it meant. There's only so many hours in the day. So this was sort of started a journey about 20 years ago to try to find more useful ways of actually linking data together. And so when we started this idea about data integration, we became very acutely aware of what the problems were in the field. And one of the most important thing that we found is that biological data really is a Tower of Babel, in the sense that different fields work around their own specialized data types. Programs especially are specialized operating on single types of data, and this was really a pet peeve of mine. We found a number of manufacturers who created instruments that would generate data files that could only be read by their software. And in fact, that was part of the entire marketing strategy. If you bought their instruments, you had to use their software. And so this basically guaranteed an income stream to the scientific equipment manufacturers. The problem is the fact that what happens when the equipment goes obsolete or the company goes out of business, you're stuck with data that you can't access in any way, shape, or form. And another underlying problem with biological data is that unlike a lot of fields in science in which you have a quantitative mathematical framework, a lot of the biological data lacks a quantitative structure. So associating a lot of this data together is very difficult. And even worse, that we found the reductionist scientists has driven stovepipe data models, and I'll give you a couple of examples of that. But one of the things that came out of this experience is something which, of course, is not going to come as a surprise to anyone who's worked for data for a long time. And that is, you really need a universal language to be able to exchange data. Right now, for example, in science, the universal language is basically English. If you go to an international conference, everyone will be using the same language to communicate. Now, in their own laboratories, at their own home institution, they can use any language they want. But when you're trying to communicate to the scientific public, you have to use a controlled language. Otherwise, no one knows what you're talking about. The other thing that we found is a common frame of reference. How does the data relate to similar data? One of the things that I've done at Pacific Northwest National Laboratory is talk to a lot of other data scientists, especially in other fields, like, for example, in the climate change field. And when I talk to the climate researchers about the problem of integrating the data, they said, why do you have any problem integrating data? We have no trouble integrating data. All we have to do is that when we have a weather station, we know we just use a GPS coordinate. So all the weather data is basically normalized to where it was taken, what time it was taken, the altitude it was taken, and at that point you can say, what is the temperature, what is the wind, what's the sunshine, all these things, and they all relate to a certain spot on the face of the earth at a moment in time. And that universal frame of reference, to be able to take all the data about where things are on the face of the earth and link them together to some common framework, has resulted in incredibly powerful software. If anyone's ever looked at Google World or Google Maps or any of these other wonderful ways of finding your way from point A to point B, it seems magic, and it's only possible because all this data has a common frame of reference. Now, of course, if you had data that had a similar frame of reference, so you could integrate it, you'd also have to know what the quality of the data is. In other words, what do you do with bad data? Well, the most practical thing to do with bad data is, in essence, not use it. And how do you know whether the data is bad or not? Well, if it doesn't make sense, if it doesn't fit. I mean, that's a sort of a glib answer, but a good example is going back to the example of Google Maps and such. When Apple Maps first came out with its software and people tried to get from point A to point B, it became very obvious that some of the data was bad because it didn't fit. But once again, you had a conceptual framework, and the data, you could judge how good it was by how well it fit within that framework. And we need to find something similar to that in biology. So we can say, oh, this doesn't fit because it doesn't make any sense in the context that it's been gathered in. Now, once you have quality data, you obviously need to be able to discover it. How do you find the data? And persistence, how do you keep it around? If you found some data last year, you should be able to find it this year. But unless there's some persistence mechanism, that's not going to happen. And I've known a number of cases in which I had some great data that I found once and can't find again because there was obviously no way of keeping it persistent. So what do we currently do now? Obviously, these are things that we would like to do, and we would like it to be completely transparent and enable us to do great things. But currently, to be able to face these problems, we have a number of different scientific databases. So everyone's familiar with the public repositories, GEO, which is where you can deposit gene expression data, and the PRIDE, which is basically where you can put proteomics data. These are public repositories which primarily contain raw data that if you're a real expert, you can get out and you can use. In order to make this data more useful, a number of investigative organizations have actually created what they call value-added databases, in which they take the data from public repository and other data that they've collected, stitch it together, and provide useful information. Like one database I use quite a bit is GeneCard. If you want to basically find any information about a particular gene, the protein, an antibody, or any part, this is really a wonderful resource. And then there are these integrative databases that basically have linked together information from a lot of other databases to provide some specialized framework for scientists. Now, these are really wonderful databases, but they are all created from the perspective of the creators who've said, you know, if I take all this data and put it together in this particular way and present it to people, they will be very, very, very useful. And yes, indeed, they've been very useful. The trouble is that this is not a sustainable model. In the news in the United States right now, there's been a lot of noise being made out of the National Institute of Health, which is finding that the grant support that it's putting out every year for value-added and integrated databases is starting to skyrocket, because every field of science wants their own specialty databases with their own particular type of data, and this is going to continue every year. And the granting agencies say at some point you've got to basically move this data out of the public database into some other framework that doesn't require an enormous amount of time and effort by professional curators to make it useful. And so there's currently a struggle with how this is going to happen. But it's clear that considering where the funding models are going, this is not going to continue. So one of the approaches that has been developed to try to encourage the interlinking and the integration of data is to develop data standards. And every year there's more data standards that people don't necessarily use, but at least it provides a framework in which certain data types can be basically put into a framework and theoretically shared. The problem with minimum data standards is that the amount of minimum data you need for even the most trivial experiments is enormous. So this was a schema that was prepared by a collaborator in trying to be able to capture the information on a particular on a Western blot. So if you run a Western blot, which for all intents and purposes is a quick and dirty experiment to find out what the expression level of a certain protein is, this would be the amount of information the standard said you had to record to be able to exchange that Western blot data with anyone else. And so most scientists would look at this and say, there's no way I'm going to put that much data into a database to be able to run a Western blot. We, to try to attack this particular problem about how much data, how much metadata do you really need for an experiment, we created a metadata system that we called IDEA. And the idea was to not mimic a pun, was to create a framework to store all the metadata on projects, sub-projects, experiments. This was actually very similar to the ISA tab format that's being very commonly used, but it actually made sense. And the idea was to be able to collect all this information in one place so that you could store your experiment. And we did sub-projects, and then we did experimental details and such. And then we found a big problem, and that is for this system to truly be useful, it had to be able to link and collect all the data we were generating at the time. And at the time, we were generating proteomics data, genomics data, high-throughput assays, Western blots. Some of the data would go into IDEA very easily, some would not. So when I tried to find information on an experiment, I'd have to say, okay, some of it's here, oh, some of it's on the lab notebook, oh, some of them in computer files. And so it turned out to be just as much trouble to find the data in the lab notebooks as it was to find an IDEA. So that the IDEA system collected the metadata, but what it did is it also added to the work that all the investigators had to do. And the investigators said, look, if this is not helping me do my work and do my job, why am I doing all this? And so we never really could get people to use a system because it did not add value to their work. So this comes up with a very fundamental question that we have to address, and that is, how much metadata do you need for your data to be useful? In other words, you could theoretically collect all the metadata in the world, but is that really required? When we basically started designing IDEA, it wasn't from the viewpoint about how much data we needed to collect. It was how much data could we collect. And that is, we never came to grips with the idea of how large the scientific vocabulary had to be able to communicate effectively with someone else. The more complex your language, the more difficult it is going to be to communicate with someone. Obviously, the more complex a language, the more expressive it can be. But before you can develop a complex language, you have to develop a basic grammar to be able to communicate at all. And this is really one of the things that's really hurt the data integration effort, or the data standard effort, is we've never come to grips with what we really need to do to communicate effectively with our fellow scientists. I'll give you an example of where this came from. And so, as in the introduction, we said that I've been doing this quite a long time, and I'm also very familiar with efforts that other people have made. So about 15 years ago, we were collaborating with the University of Washington to try to put these integrated data systems together. And one of the things that the scientists over at University of Washington were doing is trying to ask, what do biologists really want in data capture? And this was sort of a diagram that they were using. And they said what scientists really want to do is reduce the error in the laboratory by eliminating assumptions so that they have control over everything that's done, so they can repeat it. This was data repeatability and data reproducibility way back when. And so to be able to capture all this information on the systems that were available at the time, they came up with this system called Labscape. And what Labscape was an automatic recording system. They wired up a laboratory. They really did this. They wired up a laboratory so that as a scientist went through and did experiments, it recorded. They had electronic pipettors. They had electronic test tubes and centrifuges and recorders and speech capturing devices, so that as the scientists went and did an experiment, they captured all of this information. So they could tell you down to the most minute detail everything someone did to do an experiment. So once they had done this, the question then came, what are you going to do with this information? And it never really sort of occurred to them that this type of information might be useful for someone who's trying to audit, work in a clinical laboratory, but really everyday working scientists don't really need this kind of information. So okay, so this is one extreme, capturing everything. But how can you go somewhere between that extreme? And so when we started thinking about this, we realized that the amount of minimum information that you need for shareable data really depends upon who you're sharing the data with. So they sort of broken this up into 4 different groups of people. You want to share your data with you and your laboratory. These are trusted people. They need that data because they want to reproduce the experiments. Obviously, you want to do an experiment a couple of times before you publish it. You need to be able to write the paper, so you need to know how the experiment was done. Excuse me. But everyone in your laboratory, excuse me, has a pretty good idea of what your experiments are, why you're doing it, what the purpose is. If the people in your laboratory don't know why you're doing the experiment, you have a lot more of a problem than getting a laboratory notebook working. So there's some metadata you need for your laboratory, but not all. Your collaborators, people who are working with you, they need to combine your data with their own, so they have to have a pretty good idea of the experimental detail. You've got to explain it to them. You've got to communicate with them. You also need to have access to private metadata, and usually software that other people won't. Then it goes to your scientific peers. These are your friends and neighbors, another institution who basically are competing with you at some level. That is, what they want to know is what the limitations of your experiments are. In other words, what don't they do? Because what they want to do is they want to add on to your work. So they need to know where your data doesn't work and where it does work. So they need a different type of information. They also need a great deal of experimental data processing detail because they're not in your laboratory. They don't know what software you have. They don't have access to your pipelines. So they need access to enough details to be able to see the limitation of what you're doing. And finally, there's the scientific community. These are the people who basically are going to use the results of your data. And I'm actually consider myself, for the most part, a member of the scientific community. If I want to, for example, look at gene expression in certain cancer cell types, I just want to look at the gene expression numbers. I don't want to have to reprocess the data or do statistical analysis. I want the bottom line. And I also want to be able to have data in an Excel spreadsheet or a text file or something that I can put in standard software. And for this, I need the high-level metadata. So depending upon who's going to use your data, you really need to take a different approach. And so ideally, for all data, regardless of who's going to access it, you still need to be able to find it by searching for any relevant term in a browser, either at the gene level or at the metadata level. The data must be uniquely identifiable. You don't want to confuse it with what other people have done. It should be easy to work with any relevant software and easily translated by freely available software to common formats. As a good example of a company that doesn't do this well is Affymetrix, because what happened is I still have a number of microarrays from a number of years ago that I can't use because the software is no longer available. Well, the software is available if I want to spend thousands of dollars, but basically I can't use a lot of the data that I generated because I no longer have access to the data that was used to generate it. And this last point I'm going to basically emphasize, the data has to be normalized to a universal frame of reference. And this turns out to be the big unrealized important story. So I'm going to give you just a short story about how we sort of understood what you really need to integrate data. And basically this is determined to really determine what needs to be done to integrate data, to enable data sharing and reuse. I basically try to say, okay, let's try it. So I tried to integrate multiple types of high throughput data to understand some basic biological problems. What I found is data structures are best organized around mechanistic relationships, and the greatest current need to enable multidimensional data integration is adequate data formalization. Okay. Now, what we've been trying to do for a number of years is reconstruct biological systems, very complex systems level stuff. And to be able to do this, you need to know the parts, where the proteins are, with their levels, how they interact with their partners, how they change over time, and so forth. So this requires a lot of different pieces of information that you need to be able to integrate seamlessly. We have sequencing data, we have protein biochemistry, we have models that put this information together, and then we have phenotypes. We need to be able to link all this data together to create a coherent framework of what's going on. Now, there are currently a number of different ways you can link data. One is semantic relationships. For example, some semantic web architecture and standard ontologies that relate different types of information. And give you an example of this. So, for example, if you want to relate RNA-seq or microarrays or gene expression data, typically you would map this to a gene ID. And if you have some proteins, either by Western blot or ELISAs or mass spectrometry, you map it to a protein ID. Then you would resolve these gene IDs and protein IDs by a standard ontology. Well, let me tell you, this is good in principle and practice. This doesn't work nearly as well as you might expect. And this is a flow sheet that a flow chart we did in 2005 to show what was necessary to be able to integrate proteins that had an IPI number to a gene that had a gene symbol. And basically, you'd find some of them had ref-seq, some did not. Some would be present in the software, some did not. So in essence, what we had to do, we had to go through all this. It took days to be able to match a microarray data set with a proteomics data set, because in a sense, the proteomics people frankly didn't care about whether the gene expression people could integrate their data, because that wasn't what they were focused on. They were focused on the protein people, not the gene people. And that's what I mean about different fields of science, and have basically stovepiped a lot of this terminology, which is one of the reasons why we have such a Tower of Babel. So another way of doing it is mechanistic relationship. And this turns out to work fairly well. And by mechanistic relationship, I mean, as an example, if you have a genome, you can map onto the genome, for example, DNA methylation. You can map where the RNA mapped. And this is particularly good with RNA-seq, which basically allows you to sample the expression data that maps a particular gene. But the nice thing is that mass spectrometry-based proteomics also does the same thing. You map the peptides, which then maps to gene sequences. So you can map proteins or peptides or reads or mRNAs, and all this data you can basically transfer from one framework to another, because you have a common frame of reference in which you're going to be able to map it. Once you have a common frame of reference, you can use things like NoSQL databases. In our case, we took all this data, we put it into an H-based framework in which we could take all the different types of data and basically map it to genome sequences. The last one is metadata relationships, and that is a lot of this information, for example, you can't put on a genomic framework. So in this particular case, we found the most useful thing was basically sample-oriented and ISA tab formats, in which, once again, going back to the original framework that we had, project, sub-project, experiments, sample, biological context, raw data, all this is linked together in a coherent framework. And we have found that the really most useful thing is sample ID. Somewhere along here, you have to have a unique ID to be able to index this into a database. And it turns out that you can't run an experiment without having a sample. So everything in the biological sciences, the thing that really is the most unique is the sample. And so we've basically taken the sample as the framework in which we organize everything else. And then you can create from samples, you can get sample type, sample generation, sample analysis. All this information maps very nicely onto the ISATAB format. But what's different about this type of framework based on sample actually maps to the workflow rather than some arbitrary ontology. So you generate a sample, you basically analyze the sample, you analyze sample in an assay on an instrument that generates primary data that goes secondary data. So this schema, in essence, maps to the workflow. So as an example of how we use this, we did the test case of what's the relationship between mRNA expression and protein abundance. We basically did an experiment in which we basically took cells grown in a bioreactor, and in this case, we used bacterial cells grown at a high oxygen level and low oxygen levels, and then took RNA-seq data, proteomics data. We then mapped this using a HBase data warehouse, using genome taxonomy, And in essence, this exercise was done to find out what metadata did we need to collect to be able to do this data integration. From this, we created a workflow, a sample, the raw data, this is the metadata. We normalized it. We then put it together, and then we were able to do things like calculate translational efficiency of each gene. And from this type of data integration, we were able to look at different relationship between mRNA copies and protein copies per cell, both at the individual gene level and at the average gene level. And you can see very nice relationships from this. And you were able to find out the effect of data filtering, normalization. And when you added things like codon usage pattern, which of course came from the genomic data, you could build a more and more accurate model. So as things went on, we found out, well, this worked really, really well. And so the real trick that we found is that it was data normalization that was the real bugaboo. And that is to integrate protein and mRNA data, it was necessary to convert the raw data to copies per cell. But to normalize a copy per cell, you had to measure the cell number and the protein levels and the mRNA levels and the sequencing depth. There was an enormous amount of data that you had to collect in addition to the primary data to be able to do normalization. And currently, most single data protocols try to normalize between instrument runs, not between data types. And so we've been struggling with how do you normalize data. We've tried adding internal standards, like spiking in internal standards. The problem with internal standards is that the internal standards themselves perturb the experiments. And so that turned out not to be a very good one. And this is becoming really a problem, especially when you go out to public databases in which you look at data that is not necessarily normal, supposedly normal, but is it really normal? And I'll give an example of what we've come up with right here. Is this, so for example, there's a nice database out there about you using RNA-seq across different tissues. All this data has been, quote, normalized to RPKM, which is reads per kilobase per million reads. And what I did is and when we looked at this data across tissue, a lot of it makes no sense whatsoever. So what I did is I said, okay, let's take 3 housekeeping genes. These are genes which, according to most of the studies, do not vary across cell types because they're basically fundamental for life. And this is like a nucleopore complex. This is lactate dehydrogenase. This is, I forget which this one is another one. These are documented as housekeeping genes, and this is what the database says that they are across tissues. Well, they're really not very similar across the tissues. So it's not clear that if the data is not properly normalized across tissue, and you look at a really interesting gene, how do you interpret that? And so I look at this data and said, you know, I can't interpret this data. So you can't tell unless there's some standards come out that have been developed, whether this is a problem of normalization across different samples or whether this reflects reality. Well, there's a possible solution come up in the sense that that there's a conserved set of proteins and genes that really do not seem to be changing. And this shows one article, and we've actually been looking at protein expression and gene expression across different cell types. And it turns out there really is a subset of abundant proteins in mRNA that show remarkable conservation abundance across species and conditions if you do the careful data normalization. And so these kind of these molecules, which do not change across expression, really could be used as internal standards. I think this idea actually has been proposed by Matthias Mann's group as well. And we're finding out that the precision of different databases can be determined by variance analysis. So these are 3 different data sets from Kahn et al. And our data, and from Lawrence et al. About expression, about variance of a set of proteins across cell lines. And these using 3 different types of proteomics technologies. And this is log2 variance. So low variance means there's very little difference. This is the number of proteins. And you can see Kahn has the best data, ours is very similar, and the other group has a little less. This is basically a precision measurement. And these arrows here show the same proteins in this data set versus this data set. So I can look at this and say, this data set is more precise than this data set. The average is the same, but the precision is very, very different. So the other learned lesson, the essential metadata for data integration is usually generated by multiple people using different workflows. So software to capture data and metadata should be optimized for that particular task and user. Software to search and access data should be optimized for that particular data. In other words, one size does not fit all. Data capture and data sharing are totally different tasks. And until we can map the metadata capture onto standard workflows, it will always be a problem to be able to get it. So in my last slide, this is my idealized future data management system in which the institution is responsible for managing metadata on instruments, managing the raw and processed data, maintaining a metadata database, which basically take institutional project data, and then the lab group would be responsible for sample ID, data analysis, sample information, and then there'd be standardized processes to basically take this data and shove it out into data sharing sites that would basically have the information that was needed for particular groups of people. And by splitting up this into a workflow-based system and being able to capture things automatically as they're generated, I think this is the path forward to basically getting around the data bottleneck. And this is the kind of systems that we're working on here at PNNL. Well, that's sort of in a nutshell is sort of what I wanted to talk about today. Are there any questions at this time?
Thanks, Stephen. That was a really interesting talk. If any of you have any questions, you can actually post them in the questions drop-down menu. You can ask them in the chat, or if you look at your panel on the right-hand side, there's a little hand symbol. You can click that and raise your hand, and we'll unmute your mic so you can ask a question. In the meantime, I've got a question to ask you, just based on the last bit you were talking about there, Stephen. So you mentioned that you had to change how you calibrated your data based on how you were managing your data to use universal calibrators. So in systems biology, I know just making models and integrating data into models really changed how people were collecting data and also how they were running their processes. Do you think once data management's more integrated into the process, that it actually has the opportunity to improve the way different data types are actually collected for science itself?
Oh, yes, absolutely. But, you know, because in a sense, it's like once you standardize everything, then people have certain expectations of what you can do with that data. And one of the things that we have trouble is trying to convince people to put data into these systems, okay? Because they say it creates more work. It doesn't actually take away work. And so that once data management actually adds value to the scientific process, then people will start to use it. And I think that's really what the key is. Does the data management system help you, or is it an extra layer of work you have to go through? So one of the things that we thought would be very interesting is that a lot of people don't know all the things you can do with your data. So we've developed this sort of a mock-up of the ideal workflow system. Would be that here's on the right-hand side was all these list of tools you could use to analyze your data, and on the right-hand side was all the pieces of metadata you were adding to your primary data. And what would happen is as you added more metadata to your primary data, the tools that you could use to analyze it now would start lighting up. And you say, now I can use this tool. And so the more data you would add, and now I can put protein levels. Now I can do extraction efficiency. Now I can use this piece of data and that piece of data, and I can do experimental types. And now here's your new independent variables. Now suddenly all these tools now become available to you. So people would actually get the positive feedback between putting the metadata into the system and getting more use out of the data that they have in that system. But right now it's like, I'm putting this data in the system. Why am I doing this? It's a pain in the butt.
Yeah, we'd get that a lot. So I did wonder, so do you think the onus is on the software developers to incentivize data management more, or do you think the onus is on people also being able to understand the potential benefits of managing their data better to start with?
I think it's the software developers that are going to have to do it. I mean, you know, if you look at the history of all software development, and that is what drives use of something is someone who comes out with a software system that really shows the power and the potential. I remember back, you know, in the midst of time when there was no such thing as Google on the web. There was Alta Vista, there was Yahoo, there were all these other search engines. And to try to find something on the web was almost impossible. I was like, oh, you almost had to know where to go on the web to find something because the search engines were so clumsy. And then Google came out with its software tool that basically took away the problems that you had with all these other search engines. And very quickly, they become adopted. And you know, everyone, if everyone doesn't use Google, at least they use software that's actually modeled on the framework that they have. So I think the honest is that if software developers create software that truly is useful and makes a huge impact on people's scientific career, people will use those tools, and that will drive the adoption of data management standards.
Okay. I have a question from, I think she's called Anjani. I'm going to read out the question. It says, if you were given RDF and JSON data storage format for storing a disease model or any network that your software builds constructs, which one would you prefer and why? I did get told it's a very specific geek question, so.
We played with RDF. I think that RDF works pretty well, but there's a lot of overhead. I think I'd like the. Could you repeat the specific question again?
Yeah.
If I was getting data in JSON or RDF format?
Yeah, for storing a disease model or any network.
Mm-hmm.
Which one would you prefer?
I have to think about that. So, sorry, I'm going to defer the question for a long thought.
We can also get back out.
I don't like either one of them.
Yeah, we can get back to them outside of the webinar as well, so that's fine. Mm-hmm. You can have time to think.
That's fine.
Yep, we've got a couple more questions. Here's a question from Gladys, which is, have you experienced receiving already normalized data from a wet lab and not finding it necessary to backtrack to the raw and noisy primary data source?
Oh yeah, absolutely. And the thing is that one of the things that we've done is our experience is that there are certain investigators who are really meticulous, and so what we've done is we've taken data from those groups, and in essence, to see how good it is, we compare it to our own data. So we have some data sets that we've been very careful about generating, and when we get the data from other groups, the first thing we do is we compare it to ours as far as the noise, the variance, the variability, and if it looks good, we've used it, and so far we haven't been burned by that. I think it has to do primarily whether the people who are generating the data truly understand what you need to do to normalize it. Like a pretty good example is RNA-seq. Everyone says, "Oh, RNA-seq is so great because it actually quantifies reads." Yes and no. In order for RNA-seq to be adequate and data normalization has to be adequate, you have to sequence it to a great depth. You have to cover everything, because if you're only basically sequencing half the reads that are there, then in essence you're normalizing it to half half of the mRNAs, and that throws everything off. So you really have to be aware of sequencing depth and precision, and we've published some papers on this, and finding out to really get truly normalized data, you need to really go to enormous sequencing depths. Well, people aren't going to do that, so you have to find some compromises, and that's the problem. We haven't found the compromises of how do you normalize low depth sequencing data.
Okay. Okay, so I think we have just one more question for you, and then I think it's about time for closing the webinar anyway. So the next question is, in order to have good data, as in standard compliant, accessible, etc., you need good infrastructure and tools to help, but you also need quality measurement criteria for the research data. Is there anything you can say about the how to set a methodology for measuring research data quality within such a framework?
That's really hard. And the reason it's really hard is because these are areas of debate all the time. Because in a sense, there's to really truly do high quality data means you have to be dedicated to it. And that means you have to be aware of it. And people argue and debate, for example, what's the right way to normalize data? How do you take proteomics data and roll it up to various forms? So I think that what happens is that there are some standards out there, but they're not necessarily the ideal standards of how to normalize and how to really assess data quality. I think what will happen over the years is that once people start reusing data and start getting criteria out there for what constitutes good data set and high quality data sets, and this is really something that's of value to the scientific community, we'll come together and define these things and basically build into the workflow. So the expectation will be that you will normalize data in certain ways. I mean, microarray data is a classic example, is that it took a number of years before people really came to grips with what do you have to do to normalize microarray data so that it's exchangeable and usable between different laboratories. And I think you're going to have that happen with all types of high throughput data that is going to be commonly used between labs. But until people are using this data and demanding that it conform to certain criteria, I think it's going to be up to the individual research groups to set good examples.
Okay, that sounds much like what our experiences tell us so far.
That's good.
Okay. Okay, so that's it for today. Thank you very much, everyone, for joining, and special thanks to you, Stephen, for a wonderful talk and a lot of insightful answers to the questions. When you log out of the webinar, there'll be a short survey that pops up asking you some details that are useful for us to find out about the webinar. But also, if you want to get in contact with us, if you leave your details, we can also contact you if you're interested in speaking with us about data management. So once again, thank you for your time, and I will hopefully see you all at the next webinar at the end of the month. Thanks very much. Bye.